Okay, we're going to learn about our next part of our module, which is mitosis. We're going to learn what happens at each stage. So we're just going to do an overview from when we had our last section. Mitosis is that cellular division involving our non-set cells. So from our last module, we learned about what our non-set cells consist of. We learned about what diploid cells are. Diploid cells are our two sets of chromosomes. And we learned about that end result. So just to go over that again, because we're going to go over each stage of mitosis, we are going to start with a parent cell. So our parent cell, if you look again, is here. And let's say that we cut our arm and we need to replenish those cells. It goes through that process of mitosis. After it went through our interphase stage, and from our last module, we learned what each stage was during interphase. So we have this parent cell here. It's going to go through that process of mitosis. And then at the end, we are going to have two identical daughter cells. So we're going to learn today how we get from that first part of that parent cell to these two identical daughter cells. So just to, refresh our memory, we have our interphase. So before our cell goes through mitosis, it has to go through interphase. It's very important for it to go through interphase because we have to replicate that DNA and have that cellular growth to prepare our cell to divide. So it goes through three stages. So we have our G1 stage, which is that normal function. It's going through that cellular growth. And organelles are created. Then we have our S stage, which is where our DNA duplicates. So this is important because for our cell to know what it needs to do, it has to replicate that DNA. Then we have stage G2, which is it providing that further cell growth and preparing the cell for mitosis. So if we look at a cell undergoing interphase, this is how it will look under a microscope. We should know from our organelle modules that this is a plant cell because of how it is shaped in those little squares like little cells. And again, this is how our cell will look under interphase. So our first up is prophase. So this is the first stage of mitosis. This is where our chromatin, which is that DNA strand, coils. So at first our DNA is a little bit loose inside of the nucleus. When it's preparing for that first stage of mitosis, it has to coil up into chromosomes. All right, and then you will start to see that nucleus breaks down, spilling those chromosomes, having it exposed so it can properly divide. And then you will see those spindle fibers on the end of each pose so it can prepare it for that division. So if we were to look at prophase under a microscope, this is how it will look. Our next stage will be metaphase. So metaphase, the spindle fibers are attaching to the centromere. So when we went over chromosomes, we learned where those centromere was. It will attach there. The spindle fibers will pull those chromosomes into the cell's equator. So if we were to look at this under a microscope, this is how it will look. And as you can see from this slide, that those chromosomes are pulled to the equator, so it's kind of like lined up in the middle of that cell. And this is another example of that here. Next, we have anaphase. So during our anaphase stage, those spindle fibers will pull those chromatids. And we should know from our chromosome module is that our chromatids is that one part of that chromosome. So it will pull those chromatids to each side of those poles. So if we were to look at it under a microscope, you will see that our cell under anaphase, you will see them being pulled, almost being pulled apart. So when it's lined up in the middle, you will see it kind of pulling apart like that. And that's another example of anaphase. So once those chromatids pull apart, our cell then goes into telophase, which is that fourth stage of mitosis. So this is when the spindle fibers start to break down because they have performed their duties. The nuclei forms around both sets of those chromatids. And then those chromatids starts to uncoil into chromatin. So if we were to look at this under a microscope, you will see that the nuclei has reformed around both sets, and it's starting to almost look like those two identical daughter cells. So when I went over cell cycle and I went over interphase in our last module, I said that there were 4 stages in mitosis, but we have to complete this step. So we have a separate part of that cell cycle. So when we look into realization, our cell cycle will consist of our interphase, mitosis, and then cytokinesis. And when we look at our telophase under the microscope, this is another example of that too. So cytokinesis is when we are splitting those cells into those 2 new daughter cells. For animal cells, which is us, the cytoplasma is divided by pinching inward of that membrane. And when we pinch inward like that, it will form those 2 separate daughter cells. Now, when we look at plant cells, because our plant cells have a cell wall, they cannot pinch inward like animal cells. So they will form cytokinesis by forming a cell plate along the equator to separate the 2 new nuclei. So that is how it will get its 2 new daughter cells. If you have any questions, please let me know. Please continue to your next module.